And what I'd like to do is make this different than you just watching an expert on television or reading a newspaper article. I'd like you guys to become the experts. And I'd like you guys to be able to use the language that is used. You'll be able to interpret what's coming out. And instead of being sort of passive recipients of information, you'll actually understand the information. And you can then apply it to yourselves, your friends and family, or the various missions that we have with the church, or in your life with social justice and otherwise. So anyway, I hope that I really don't want to make this too technical, but I'd like it to be a little bit like learning another language. And when I went to medical school, they basically said, listen, all you're doing is learning a language. And, and we can act so important because the language it's basically Latin. And so I'm calling this Babel Scientific, the COVID edition. And I would love us, to start talking back and forth. Eventually, I think it'd be really interesting if the people in our community who've actually had COVID might come in and tell us their experience. And the reason that might be so interesting is this is a disease that's just being described, and their experiences are as important in understanding the disease process as anything else. So I also would like us to be able to be good at seeing when misinformation is coming our way. And then this is a field where, or a problem, an issue where a lot of disinformation and conspiracy theories are being developed. And to the extent that you guys are really good at the language, you can, and then maybe the information I can get for you, we can start to dissect those. And maybe we can, I don't want to do it now, but maybe we can tiptoe into that area and start to be able to, be a place where real conversation can occur. And I bring that up because I just want to go through real quickly. I'm scared off by it. So, as I said, I want this to be Babel Scientific, the COVID edition. And, you hear all these agencies listed, and one may not even really know what they are if you think about it. And so there's the Center for Disease Control, and that is located in Atlanta, Georgia. And the reason I want to bring up the agencies is just for you to realize that there is, within the health profession, there are issues of public health versus treating the individual. And sometimes there's a tension between the two, because what's good for a population or a larger community may have problems for an individual. So for instance, we may treat something for the population that reduces the incidence of something. But as a complication, you get a few cases of autism. Well, for the population, that's pretty good. But for the individuals that get the autism, not so good. And there's always a tension between the two. So I just want to point out that the Center for Disease Control is really a public health. It's an agency within our federal government for public health. The National Institutes of Health is what funds our basic science. And, they're who generated the vaccines. What a scientist wants is usually for the advancing of health, but sometimes there's a tension between the scientists and the population. What they want out there may not necessarily have been tested enough. Sometimes there's been unethical things done, and we are evolving as a civilization to know how to regulate that. But I just put that in and then the third thing is where the individual is sort of addressed, and that's with the Food and Drug Administration, or the FDA. And they're tasked with approving our medications and vaccines. And I just sort of target it as being where the individual is protected against. But, there are tensions between these three groups, and I think we just need to understand that they're there. Where I want to go next is I just want to talk about this, a cell, a generic cell, real quickly. And I don't know, here's my generic cell. And a cell is going to have a cell membrane, it's going to have a nucleus, and it's going to have a cytoplasm. The genes are in the nucleus, and the genes are made of DNA. The DNA is copied. A copy is made of a gene in the DNA, and it goes to the cytoplasm as something called mRNA. And the mRNA makes proteins. And our body works by proteins. And it basically drives our whole body works on proteins that are made from genes in the nucleus. And so about DNA, it's a double helix. What that means is it's just two strands. It splits apart. A copy is made out of it. It goes here, and then a protein is made. Now, the reason that is relevant here. So, and the building blocks of a gene are nucleotides. And so there's some, and and then the building blocks of proteins are amino acids. And the building block of proteins are amino acids. When our cells replicate, they reproduce the genes. When the gene is reproduced and a mistake is made with one of the building blocks, that is called a mutation. And so then when a copy is made to make a protein, it makes a protein that is different by a single amino acid. So it changes the protein. That's what the mutation does. And we have been hearing a lot about mutations lately. And and the reason I want to talk about this is now, let's talk about a virus. How does a virus work? A virus is different than all the other types of, I list on the outline just the types of cells that exist in the world. We know about bacteria, parasites.
Claudia, can I make a suggestion? If everybody would go to speaker view, you will see her diagrams bigger. So in your upper right corner, if you don't know how to do that, where it says view, click on that and go to speaker. You'll see her diagrams bigger. And then if Claudia, you want your outline back up, we can do that after your diagrams. Thanks.
In the world, there are, and they go along the evolutionary scale from not complicated to very complicated. And we go from bacteria and up that ladder to parasites, fungus, plant cells, and then animal cells. And they all sort of have that general idea of genes being in the nucleus that has DNA that then has mRNA. It's called messenger RNA. It's a message as to how to make protein, goes into the cytoplasm and makes that protein. Now, the reason I just put these lists of cells is I want to tell you what a virus is, because it's pretty bizarre. A virus is basically a set of genes wrapped around in an envelope. And it doesn't have the machinery for anything. And what it does is it binds to a cell, and it's like a lock and a key. I mean, a key and a lock. And you hear about the spike protein. It has on its outer surface, like a key, the spike protein. It then uses a spike protein then into a lock, or to kind of like a dock also in an animal cell. It attaches and it gets swooped into the cell, and then it hijacks the cell's machinery to reproduce. So, and it's really interesting because it's rather sophisticated. It uses machinery and genes that are as sophisticated as a human cell, but it basically has none of the apparatus of it. The way we have fight viruses, so the way we have been getting immunity from the coronavirus is generating antibodies to the spike protein, which I've drawn here. And this is the receptor on the human cell that is like the lock. And we generate antibodies against that spike protein, and it covers it so that it no longer can go into the lock. And that's why it's called a neutralizing antibody, and that's how it works. The problem with all these variants is that a protein with its different building blocks or amino acids takes on a shape, or what we call a conformation. And that's where the locking, the key and lock is so important, is there's a certain conformation or shape, and it has to fit perfectly into the lock. And if it doesn't, then it doesn't work. If it starts out not doing a good job doing it, and through the mutations, it actually starts to do it better and better, then it becomes a virus that's able to take hold more. And so with what's been happening with these variants that have come up is that as a virus reproduces or replicates, mistakes or mutations happen in the reproduction of the virus's genes by the host cell or the human cell. And anytime a mistake has been made, we kind of refer to it as generating a new strain of the virus. If the strain is less good at connecting through the key and lock, it's just going to die out. But if it's better, it's going to take hold. And the terminology, the idea is it goes from a strain to the lingo is to call it a variant if it's gotten better at it. And our various variants right now seem to be variants that make this key and lock work better. And that's why it's increasing transmission. Now I want to get to talking about the various vaccines. They're referring to them as different platforms for the vaccines. And generally, we have either used the protein and isolated the protein and have injected that into human beings, or we use a form of the virus, like polio virus, for instance, that has been changed somewhat so it's not as what we would say virulent. It doesn't cause as bad disease, and you generate an immune response to it. They are testing right now a new platform, and you hear it referred to as the mRNA platform. And that has never been used before as a vaccine. It has been being used in cancer treatment, but it has never been used in as a vaccine. Now, the way the mRNA works is it's literally what's delivered in the vaccine is the actual mRNA that I told you goes into the cytoplasm to make the proteins. Because of the Human Genome Project and us being able to sequence and make genes, we can make that mRNA look any way we want. And that's why getting the sequence from China was so important. We got the sequence for the coronavirus that turned up in China. And it's really important to realize this is not the mRNA for the whole virus. So you're not making virus. You're simply making it for what they are making the mRNA to code for. And it is the spike protein. So then, and so once our cells are making the spike protein, it then gets in your blood and you start making antibodies to the spike protein. And it goes to, and then it acts as though you've been infected with the virus. Antibody covers up the spike protein and it can't, the virus can't bind to the human cell, it can't infect it. And that's how you get immunity. And the Pfizer vaccine and the Mrna vaccine are made from mRNA. These are the ones that have to be kept so cold. And the reason they have to be kept so cold is for the mRNA is a little bit unstable, but these get wrapped in what is called a liposome. And it's a lipid, a fat material that RNA has to get into the cells. So it has to cross the membrane from the outside going in. And it uses this little covering called the liposome to get in. And that all of that has to be kept at a very cold temperature or else it falls apart. So those are the that have been out first and they've been approved, which is interesting, even though we've never done vaccines using this technology. The next technology, it uses what I'm going to refer to as a vector, but it uses a different virus. And by molecular biology or recombinant techniques, recombinant biology, you've heard those terms. It means we take a gene and we recombine it with genes from a different organism, and then we form a new one. And that's what we're doing with corn, and that's what the GMO stuff is. But with this, what they've done, Johnson & Johnson has taken the common cold virus, and it's called adenovirus, and they recombine it with the gene from coronavirus, but the gene is the spike protein. And then it goes and it has its own docking protein or key and lock. It infects the cells. You make adenovirus and antibodies to that, but you also make antibodies to the spike protein. And those antibodies work in the same way. Johnson & Johnson's vector is adenovirus, which is the cold virus. AstraZeneca uses a chimpanzee virus. And it literally is an infection. And then the last one, the test tube, or a large test tube, they actually use techniques to have cell cultures. Cells that are in culture in the laboratory produce large amounts of the spike protein itself. That's purified, then that's injected into you. And you make antibodies against the spike protein. So all of these in one way or another make antibodies to the spike protein. And the thing that we can do with the mRNA vaccines is as a variant emerges, we can very rapidly alter the RNA to be the exact same gene that is in the variant. And if the vaccine that was from the previous vaccine isn't working any longer because the spike protein was so changed, we can just change the sequence. And that is what they're actually starting to do. So here's the deal. As you're hearing this, and now I'm going to step away from all the scientific talk, it becomes a little bit harrowing as you really think about what we're doing. And, I sort of was telling you the hard facts of what's happening. We've been doing this all along with the flu vaccine, and in a way, accepting it. Whether there are safety issues with all of these is really something that, we need to discuss. And that's, of course, why the large scale studies have been done before they're introduced into the population. Whether that actually picks up the complications that can come from any of these vaccines is something that time will tell. And so I'm going to stop here. We can talk a little bit further about the efficacy of these different ones. We can talk about how long the immunity lasts. And we can talk about, different things that emerge from that. But this is a moving target right now. And it brings up so many issues. And, I'm really happy, I'd love us to be able to have a discussion about this. I do have people that I can talk to where if I can't answer the question or you don't feel that my answer quite brings in the issues that you're interested in, I do have access to people, a range of people who I think are kind of interesting that could, maybe, fill things out. So that's my introduction. Does anyone have anything, where would you like to take this next, Jean? Do you have any suggestions, Jean?
Well, I saw a hand, so why don't let's go back to gallery view for a little bit so we can see everybody. Hi, ABX. I'm not sure who you are. My name is Jean. Hi, you're on mute. So.
Hi, I'm Andy Brown. I'm an ESL teacher, and actually I'm from the Caribbean, and we'll be having AstraZeneca. So, yes. Claire, I really want to know about this. What's going on? Yes?
Wonderful.
Yes.
Okay. Hi, Andy. Welcome. This is my friend all the way in the Caribbean, Jamaica. I said is joining us.
Oh, hi.
Welcome.
Yeah. Okay.
Welcome.
Yeah.
Right.
What I know about the AstraZeneca is it's a platform that has been being used, and we have all been getting it in the flu shot. And so I haven't, in a way, thought to ask the question that I'm now asking about the flu shot. And so the AstraZeneca, and it can be kept at room temperature. Now, it is requiring 2 doses, but there doesn't seem to be safety issues around it that we are concerned about. What the problem that's emerging about AstraZeneca is the sequence that they're using for the spike protein doesn't seem to protect as much against one of the variants that is emerging from South Africa. And so they're actually pulling it from South Africa. Mm-hmm. And it can't be, because of the type of platform, it can't be altered as fast as the mRNA ones. The mRNA ones can be altered at the drop of a hat. In a relative sense, there's nothing wrong with it. And if you can get the idea is if you can get the population vaccinated fast enough before that variant takes hold, then it's safe and effective.
That's the big question. If
I think India's stopped it too. India says it's not going forward.
Where?
AstraZeneca in India.
Yeah, India.
Yeah.
And yeah, and I know that India is involved with the World Health Organization, and they are their vaccine that they're developing is a soluble protein. But that that it has the conformation, the structure of the first the one that arose in China, and so it may end up having a problem too because it may have the wrong conformation or shape. So it's interesting with physicians that I've been sort of all interacting with, we were all like, oh, we're not going to get the mRNA one. That sounds too, I don't know. And we've all sort of evolved to thinking we'd like to get the mRNA one because it can be altered to get
sequence
variants. And, when we really thought about it, we didn't really like being infected with these altered viruses. But nobody really knows, and you end up having to balance, do I want to get COVID or do I want to take a vaccine that has largely been shown to be safe?
Sure, always.
Does anyone have another question that you want to ask? Daria.
Yeah, you mentioned the chimpanzee virus.
Yeah.
Now, that doesn't sound safe to me. Like, is there an issue with that?
Well, you've had the flu vaccine. You've probably had a chimpanzee virus infection. This is what they use in the flu vaccine, believe it or not.
So it's okay.
It's okay. It's okay. I mean, we think it's okay. It hasn't, and we need to follow these studies. Studies are important in terms of, from an equity issue, we need to make sure the population it's being applied to has been actually studied. And very often, people of color have not been included in the studies. So we don't often know safety. And that's what in that particular population. And that's one thing that social justice advocates have been making sure of, is that the study populations include people of color from particular characteristics. Yeah, Fred.
Well, I guess I've given up. Well, I understand what you're saying. So we learn the language so we can talk about it and work with our friends or relatives but I pretty much have given up on people who don't take this serious enough, or they're going to wait forever for the final results of the vaccines to see if anyone drops from it before they take it. And they're probably going to be the people that keep this virus going. And they're saying now, I read that the people that don't get it are going to be the ones that are going to be passing on the variants and keep that variance going.
Well, Fred, you have it exactly right. And, how Trump and this guy that was in Atlas, the guy that ended up advising him and wanted herd immunity. Well, these mutations only show up if they can replicate and take hold. And that's where a mutation occurs. And then it generates a variant. So, for instance, there's some argument as to whether we should give the second dose of the mRNA vaccines, the Mrna and the Pfizer one, because you get pretty good efficacy without it, and we need to vaccinate more people. Well, the reason Anthony Fauci is saying no to that is no, we need, whoever gets vaccinated, we need to have the highest amount of efficacy so that no replication occurs, so that we don't get the appearance of, we don't give the virus a chance to have the variants appear. And so the same thing applies, Fred, to what you brought up. If we have people where it's replicating in, and it's a small number where the variant is going to rise, and then the people who have been vaccinated, if that vaccine doesn't cover that variant, then it's just as though you weren't vaccinated.
Oh my gosh.
I know, Jean, but I think I see, Cleo, and then perhaps I would like to address that. Yeah.
Mhm.
Oh, Cleo, his hands is up. I think Jean is seeing her.
Um,
Oh,
Should I go, or Jean, do you want to go?
go ahead, Cleo.
Yeah.
I just had a question about two vaccines that have to stay cold.
right.
That is the Moderna and the Pfizer,
Yeah, and the mRNA one.
and the mRNA one. Are
Well,
those
they're both mRNA.
those are the
Right,
mRNA?
right.
Now with the variants, I think there's another like Johnson and Johnson that has a pretty good efficacy, like 85% maybe coming out. What I was wondering, and you only need one dose, and you don't, it doesn't need to stay cold. Is that, do I have that
So,
right?
so real quickly, I'll try to talk about efficacy. Efficacy, you can think of efficacy in many different ways. Will you ever get do you get the infection? Or if you get the infection, are you going to get sick? And if you're going to get sick, is it going to be mild or is it going to be severe? So when the numbers first came out, there's this very high efficacy with the mRNA ones, the Moderna and the Pfizer. And it was lower. It was like 65 to 70% with the Johnson and Johnson. But that higher number is, and that is referring to actually infection. So these mRNA ones were pretty amazing with the efficacy. You don't even get infection. Whereas the lower number with the J&J one is you still get infection, but you don't get severe disease. And it can be kept at room temperature, or it doesn't need the very, very cold environments. So that's why it's being used more broadly. And as the individual, and this is where with time, we can maybe discuss this further. What number do you want to go after? Do you want to go after the very highly effective one? Or you want to, where you get no infection? Or are we willing to tolerate one that has a little lower efficacy, but you won't get severely ill, and it can be kept at room temperature or in the refrigerator?
Are they even using those here? Now I haven't heard much of them using it. And also, is it easy to change that if it's not an mRNA vaccine for the variants that are coming out?
So the AstraZeneca hasn't been approved yet. And the only vaccines that have been approved is the Moderna and the Pfizer.
Oh.
The J&J one is going to be approved probably by the end of February.
Okay.
You can see that it has these different platforms. It can be altered, but not as easily as the mRNA one. Carol?
Yeah, the government probably doesn't care which one you get because they want us all to be vaccinated, first of all, right?
Yeah, I think we should all want to make our own individual decision.
Right or wrong?
Okay, yes, could I? Oh.
Yeah, go ahead, Maureen, please.
Yes, well, first I want to say about the, and I want to address this as a person who is to be one of those persons who will go through, take the assessments of the different types seriously, because I already have autoimmune disease. And so I think before I take it, for example, I have to see my neurologist, several other things. So for me, the idea of introducing something that is going to already prompt my body to produce systems from a body that is in a body that is already reproducing and attacking itself makes me really follow the science. The second thing is a lot of the testing was done on people of color first and foremost. A lot of the testing was done in the Amazon. It came over.
I don't think with these, I mean, in vaccines in the past, but I don't think.
No. I think coming out of Boston, they had already started done some of it already. A lot of it was done, and in fact, there was a big question about it because they're saying if it is already being done on people of color in the South, then the people of color should have equal access to it, and that is part of the argument. I think fundamentally, like the human cells, right? Between a black person and a white person, the human cells itself is a cell beneath the skin color. It's the same, right? Right. The thing is that some populations have particular genetic propensities, right? So we know like European Jews tend to have things around cancer. People in Africa have issues around sickle cell, right? So I think that is only in as far as a race things go, I think. So I think that when we're approaching it, because there are all these different reasons and all these things, it's kind of dangerous to say the problem is going to be those who don't want to take it, because you can still take it. From what I heard from the mRNA or what I've been reading with the doctors, you still can be asymptomatic and pass it on. So there's also the possibility that people who have taken the vaccine become very lax and think, okay, I've taken it, so it's just like the flu, I don't have to worry. Well, you have a second infection or you're asymptomatic and you pass it on.
Well, and so that's why I was where we were talking about the efficacy. You can still get the infection and you can still transmit it, but you're not going to get severe disease. It doesn't mean that you stop doing the mitigation procedures or until the overall population, the level of the virus has gone down to a much lower level. So I agree that racial differences in what a human cell is not different, but there are different propensities. And for equity purposes, you want your population to have been studied and the adverse events from a particular intervention to be known as much as any other population.
Because, my concern is then, because in terms of the politics of it, sometimes when the disproportionate whatever distribution, then we start demonizing, attacking the people who decide that they're waiting and that they are the cause for which so the variant started in UK and they spread it to South Africa, but now it's only called the South African variant, where it started in the UK.
No, it didn't. But they're different. But we have plenty of variants here. It's not that anyone, but I do want to say something about misinformation and disinformation. The Black population in the United States has been unethically experimented on through the ages, and there are many legitimate reasons for distrust to exist. It should be addressed, and we should be expected to address it. And if people do not feel comfortable taking it because they haven't yet had their fears addressed, that should be honored. Where I'd like us to be able to, Maureen, I'm really glad you're bringing this up because it's the kind of discussion we should be able to have. And there is a difference between legitimate concerns and then concerns that are coming from disinformation. And what's interesting, and I'm going to say a statement that some may not agree with, but what has been interesting is from what one might imagine as a conspiracy theory, it's not coming out of the Black population where there's legitimate reasons to have concern. And in fact, interestingly, is coming out of the white population. So, some people may disagree with that, but that's.
Well, I'll ask my question really quickly. It's just a matter of fact. I had the vaccine, the Mrna vaccine on a Friday. I was fine for 5 days, and then I started 3 days of vomiting in the afternoons. And I was interested that it would have been that long. Can I count that as a side effect? Does that sound right?
I don't know, it'd be interesting, and the FDA should have a place to report adverse effects. Mm-hmm. The important thing, and I'm learning this, and the people that I am interacting with are now educating me on the flu virus. And they go to their pharmacy or wherever they get it, and they've decided which platform they're going to get. I may start doing that, and maybe down the line we could start talking about that. But this isn't an infection, it's the developing, and so you might be expected to get that if it's one of the platforms that uses an infection, but not so much with the mRNA, but who knows? I mean, I don't think we actually know. And it is a foreign protein. It's not a human protein. The spike protein is not a human protein. And so you're generating antibodies against a non-human protein. So who knows what the side effects are?
Who knows? And the lists on the internet about side effects include nausea and vomiting. So,
And the adverse effect with the second one is probably going to be more than with the first one.
No, I'm just going, when you said it's going to be worse. I had my first Moderna also. I have had a lot of other illnesses in the past. I'm like 100 years old, not quite. And all those illnesses I've had, I think, changed the way the shot is affecting me. I had injuries in my arms. I had reaction in my arms after I got the shot. I had more pain, which I hadn't had for a long time. So I think it depends on who you are, what's happened to you before you did the shot.
That's a good point.
The other thing that is, I told you that it's wrapped in this liposome, and the liposome is what is the proprietary part of it. That's what the companies have spent billions of dollars developing. The reaction actually could be to that liposome envelope and not even the vaccination. And they're only separating out those two things right now.
And Abby has also had her hand up.
I think you might have answered this. Once you're vaccinated, with the 2 shots and the waiting period, then you're not necessarily unable to transmit the virus to others. And does that also apply to the flu vaccine? If I get my flu vaccine, does that mean I can't give anybody else the flu?
Transmissibility is being studied, and really, I'm really pleased with the way you guys are talking. This is really great. This information is coming out right now. And so for the efficacy for the mRNA ones, they think it's 95% for actually getting the infection. The thing is, we don't, and so if you don't get the infection, you can't transmit it. The thing is, no one knows how long it lasts. Now, with the ones that use a viral vector, you can still get the infection because its efficacy for that is a little bit lower, but you don't get severe disease, but you still might be able to transmit it. But it may last longer because it's an infection, and it's this viral vector, and it may have more durability. So, we have to be good consumers, and I would really like us all to be, these are very nuanced conversations that we need to be having rather than being blind consumers of, ripping, showing your arm and going out and getting it.
So you're gonna we're gonna do another round next week, right, Claudia? Because time is
time is over. I think I wanted next time. What I'd like to talk about is just describe what the disease actually is, real briefly, and then I'd like to talk about the mitigation strategies and what works better than the others, how to deal with the the mutants or the variants, and then what's coming out for treatment. And because actually that may be relevant. And then also what we mean by surveillance.
What
Also,
about masks?
long-lasting effects? Long-lasting effects also,
Exactly.
please.
Right.
Could you bring up masks next time?
Yeah, I wanted to make that the whole thing about prevention. Decided to go after the vaccine. In a way, if you do the prevention first, but I wanted to get to the vaccine really quickly because you guys are all needing to make decisions about that right away.
Claudia, this is wonderful. Thank you very much.
This has been wonderful.
Yes.
Absolutely.
Thank
Very
you,
useful.
Claudia. Yeah, very.
Well, good and thank you.